presenter is Deka. And yes, here to represent Deka is John Mom, who is also going to be a panelist in our second panel. So John, thank you for launching. You're good to go.
Excellent. Thanks, everybody. Thanks, SAPA. Thanks, everybody, for joining us this morning. I'm John Mom. I'm the CEO of Deka BioSciences. So here at Deka, we've actually developed in-house a versatile platform that we can target any two interleukins into any tissue. What we've also done is to develop precision patient selection assay systems that we have found genetic signatures associated with response to different interleukins. So that enables us to select patients for our clinical trials. Sorry, guys, I tried to progress. Hold on one second. Okay. Sorry about that. Looks like I'd already been practicing this morning. Sometimes it pays not to practice too much. So we're a team of highly dedicated professionals. We've been in this space for about 115 years collectively. We have experience across biotech, pharmaceutical, business development, research and development, as well as manufacturing. We plan on actually manufacturing our own molecules for phase 1. Here's essentially what we've got. We have an ability, a platform to couple two different cytokines on either side of our targeting vector. The whole point is that interleukins, cytokines work much more effectively in combination rather than just individually. So as Jeff had said earlier, it's all about stimulating the immune system for oncology. It's also about inhibiting the immune system for anti-inflammatory disease. So we have a pair of interleukin-2, a normal interleukin-2 with a high affinity IL-10 for oncology that we can target into whatever cell surface protein is present in the tumor. And we can use interleukin-4 and a low affinity IL-10 for targeting into inflammatory disease. We've raised $2.5 million. We've built our platform. We've filed significant IP. We have our first patent already allowed, and we filed IP on the unique genetic signatures associated with response or lack of response to a given cytokine pair. The idea for this actually came from the fact that cytokines have shown function in cancer. In particular, interleukin-2 and interleukin-10 have both shown about 25% response rate in renal cell carcinoma, and RCC is known to highly express the EGF receptor, as are other cytokines. The unique thing that we found is that IL-10 actually tames the toxic inflammation associated with IL-2. The other component is the combination of two cytokines. These two cytokines uniquely stimulates in a very potent way the anti-tumor response by the immune system. So we can target this into any tissue. The very first place we're going is renal cell carcinoma. What we plan to do then is to use our unique assay system and genetic signature in order to select the patients most able to respond and conduct our clinical trial in those patients, both to ensure from a safety perspective that we're treating people that we can really understand what the safety looks like, and really from an investor perspective, our clinical trial readout will actually be in people that are most capable of responding to the therapeutic combination. Similarly, with anti-inflammatory disease, both IL-4 and IL-10 have shown potent suppression of inflammation associated with psoriasis, and IL-10 has been shown to suppress inflammation in Crohn's disease. The combination of these two cytokines together and targeting into tissue then permits us to suppress monocytic inflammation, which is a primary inflammatory source for most of our inflammatory responses, but also with IL-4 to shut down T-cell mediated inflammation. So the combination of these two molecules together seems to push our effective concentration of maximal suppression to about 0.01 nanograms per mil. Cytokines, interleukins are very potent, so our cost of goods is correspondingly low because we don't have to treat patients with such a large volume of the material. The other thing that we found very interestingly is that Crohn's patients, and we think psoriatic patients, are very low producers naturally of interleukin-10. So when you stimulate them with a pro-inflammatory stimulus, they produce very low levels of this known anti-inflammatory cytokine. So the idea we have then is to find the patients that are the lowest producers and reconstitute natural IL-10 anti-inflammatory function within the tissue of interest. So we have, again, the ability to select the most likely to respond patients. We've raised $2.5 million. We've secured our platform. We've filed significant IP. We have also established our precision selection assay system. What we're looking to do is raise $20 million in 2 tranches. Tranche 1 will enable us to drive 2 molecules, an oncology molecule and an anti-inflammatory molecule, to IND readiness within the next 8 to 15 months. And then the second tranche will be used for a phase 1 clinical trial, either in oncology or in psoriasis. Again, I'm John Mumm. This is Deka. Thank you so much for your time.